Ghrelin levels generally increase prior to a meal and decrease after meals. Restricting calorie intake when dieting, for example, leads to large increases in ghrelin levels, which then increases appetite. In other words, if you restrict calories in order to lose weight, then as a result of eating less, levels of ghrelin, an orexigenic or appetite-inducing substance, is increased, while two appetite-suppressing substances are decreased. Can you see why depriving yourself of food is not a great weight loss plan? Interestingly, following gastric bypass surgery, ghrelin levels are greatly reduced, which may explain the success of gastric bypass over diets in achieving and maintaining weight loss. Next to air and water, food is without a doubt the most valuable resource we have. It fuels our thoughts, movements, and all our body systems. It helps us to grow, it brings us pleasure, and even provides a conduit for strengthening social relationships. Humans' relationship with food is fascinating and complex, and raises some interesting scientific questions, like what drives us to seek food and eat it? Why do we seek particular foods? And how do we know when to stop eating? The answers to these questions all involve physiological processes in the brain. Hunger starts in the brainstem. Since eating is essential for life, it's not surprising that circuits involved in hunger and eating are located in the brainstem, evolutionarily the oldest part of the brain. Increased activity in two areas of the brainstem's dorsal medulla, the area postrema and the nucleus of the solitary tract, produce hunger. Collectively, these regions are known as the AP-NST. Both of these areas receive information from the stomach, the duodenum, which is part of the small intestine, and the liver via the vagus nerve. The AP-NST region also contains nutrient-sensitive receptors, especially for levels of glucose or sugar in the brain. Activity in the AP-NST is associated with activation of the sympathetic nervous system, which directly stimulates the release of stored nutrients from adipose tissue. In other words, the AP-NST region signals a need for fuel from a snack, meal, or from long-term fat storage in adipose tissue. The hypothalamus region is involved with motivational states, including fighting, fleeing, mating, and feeding. Scientists have known for many years that lesions in two regions of the hypothalamus, the lateral area and the ventromedial area, are involved in hunger and satiety, respectively. More recently, researchers have learned a great deal about the roles that specific peptides, secreted by nuclei of the hypothalamus, play in regulating hunger and eating behaviors. The recent findings indicate that hunger and satiety involve complex interactions between neuropeptide communication systems, rather than specific hunger and satiety control centers. It's released by the lateral hypothalamus, orexin and melanin-concentrating hormone, or MCH, stimulate hunger and lower metabolic rate. The lower our metabolic rate, the more slowly we burn calories. These peptides are classified as orexigens, which means they are appetite-inducing substances. Orexin is involved not only in the control of hunger, but also in the control of sleep cycles. Feeling sleepy after a big meal may be related to eating-induced changes in orexin activity. Axons from orexin and MCH-secreting neurons in the lateral hypothalamus send information to a variety of brain regions, including the cortex, periaqueductal gray area, the reticular activating system, the locus ceruleus, the thalamus, and the autonomic nervous system. This rich network of connections may help explain how hunger and eating behaviors interact with our alertness, sleepiness, mood, and stress levels, and why terms like comfort food and stress eating have become part of our everyday language. The orexin and MCH secreting neurons in the lateral hypothalamus receive information from another hypothalamic nucleus, known as the arcuate nucleus. The arcuate nucleus contains neurons that secrete neuropeptide Y, also known as NPY, an orexigenic substance that induces ravenous eating. Ravenous eating is often referred to as binge eating, or rapidly consuming large amounts of food. Levels of hypothalamic NPY are directly affected by eating behaviors. Eating lowers levels of NPY, and starvation increases levels of NPY. Neurons in the arcuate nucleus directly project to MCH and orexin neurons in the lateral hypothalamus. Another orexigenic substance, known as AGRP, which stands for agouti-related protein, is also released by the arcuate nucleus. The arcuate nucleus receives input from the brainstem medulla and direct input from the stomach via another orexigenic substance, ghrelin. Ghrelin directly stimulates the arcuate nucleus NPY neuron, which makes us feel hungry and seek out food. There are substances that have anorexigenic or appetite-suppressing effects. Surely you can see how the eating disorder anorexia got its name. These anorexigenic substances include leptin, peptide YY, known as PYY, cocaine and amphetamine-regulated transcript, known as CART, and alpha-melanocyte-stimulating hormone, alpha-MSH. Let's explore the activity of the anorexigenic substances. Leptin is a hormone secreted by nutrient-rich adipose or fat tissue. Adipose tissue is a source of long-term nutrient storage. Leptin suppresses eating by inhibiting neurons that secrete the orexigenic peptides NPY and AGRP in the arcuate nucleus of the hypothalamus. Leptin therefore signals the level of nutrients stored in fat tissue. Theoretically, high amounts of stored fat should result in decreased food intake. Orexigenic substance PYY is produced by the small intestine. Amounts of PYY are directly proportional to the amount of calories ingested. PYY inhibits the release of NPY and AGRP from the arcuate nucleus of the hypothalamus. The arcuate nucleus secretes two anorexigenic peptides, CART and alpha-MSH. Interestingly, the orexigenic substance ghrelin inhibits CART and alpha-MSH. In other words, the orexigenic substance ghrelin not only stimulates appetite, but it inhibits two anorexigenic substances, CART and alpha-MSH.